So, greetings my dear students. I am Dr. Monica, working as Associate Professor in NIET Pharmacy Institute, which has been affiliated to Dr. APJ Abdul Kalam University, and the subject I am going to take is Pharmaceutical Microbiology, having code BP 303T. Now, today we are going to start with unit 4 of your syllabus, that is code BP 303T. Now, what we have to study under unit 4, we have already covered unit 1 in detail, unit 2 in detail and unit 3 in detail. Now, we are moving towards the unit 4, and in unit number 4 actually is based on 2 modules we need to cover under unit 4. Now, what are those 2 modules which we have to cover is, first is you have to study aseptic techniques. Now, how we follow aseptic techniques, what is the layout we can design, how we can design our microbial lab, how we can design a aseptic room within the industries. So, you need to study about the design of aseptic layout, you need to study about, that is, what are the various sources of contamination within any of the laboratory or within any of the area, what are the main sources of that is contamination, microbial contamination within the environment, that we need to study, as well as we also need to study about that is what is clean area classification. So, this is the module 1 which you need to cover. In module 1, these mainly you have to study, that is aseptic techniques, you have to study, that is laminar airflow, you have to cover sources of contamination of microbes, and you also need to know about the clean area classification, that how on the basis of particles contaminated or based on microbial contamination we can classify the clean area. So, this can be covered under module 1. After that comes the module 2, that module 2 will deal with the microbiological assays, how to find the concentration of unknown samples within the antibiotics, how to find it out the unknown concentration within the tablets or antibiotic preparations. So, that will come under module 2. I am going to take module 1, and in module 1, in this lecture I will mainly focus on the aseptic area. Now, what we actually mean by aseptic area, I will focus on designing, how we can design the layout of an aseptic area based on terminal sterilized product as well as based on in process sterilization. So, these 4 things we need to cover in this lecture. But along with that, first of all I would like to tell you what is aseptic. What is septic? Septic means free from microorganisms. Now, aseptic technique is a set of routine measures which we take so that we must be able to prevent our environment, we can make our environment free from microorganisms. We have to take routine measures, as in if you have seen in our homes also, we always clean with the help of disinfectants, we always use phenyl, we use Lysols in the home when we are cleaning. If similarly in the labs, when we are working with within any industrial area, we have to follow the sanitization conditions, we have to make sure that our industry must be clean, our area in the lab which we are working, that lab must be clean, that must be free from microorganism. Nowadays, we are taking extra precautions, even washing hand time to time, we are we are putting on mask on daily basis. So, these are all the techniques by which we can prevent contamination, we can protect ourselves from the microbes, we can clean our environment. So, aseptic technique is a set of routine measures that are needed to be taken to prevent the cultures which we mainly use in the microbial lab, because nutrient medias, nutrient stabs, slants, whatever nutritions, as they are highly required for the isolation purpose in microbiology lab. So, those cultures must be free from microorganisms, sterile media need to be prepared. We have to clean our surfaces on day to day basis. Whenever we are going to the laboratories, we have to put on the lab coats. If you know, whenever we are going to the laboratory, we have to follow all the hygienic conditions. We have to wash our hands, we have to cover our hairs, we have to cover our, we have to use the mask. We have to take care of each and everything, like from surface cleaning, from environment cleaning. You have to take follow personal hygiene, you have to sterilize all our equipment, you need to sterilize all your glasswares. So, you have to sterilize even the inoculating loops which are being used for the that is practicals parts. So, in the practicals, whenever you are going to the practical laboratories, what you need to do? You have to follow aseptic aseptic conditions, we have to follow hygienic conditions, we have to disinfect our surfaces wherever we are going to work, that those surfaces must be clean, and that those surfaces are always cleaned with the help of various disinfectant. How we can clean our environment that our environment in which we are going to work, that must be free from dust particles, that must be free from microorganisms. How we can clean that environment? We can clean that environment with the help of various UV lights. If we put on UV lights within the hall or in laboratory where we are working, we can clean our air, we can disinfect our air. We are chemical sterilization, you must be knowing the various techniques of sterilization. We can use chemical sterilization, we can go for physical sterilization, we can go for radiation sterilization, even by the help of fumigation techniques. Fumigation technique is also being used for the sterilization purpose. So, aseptic area is an area that is free from microorganism, and for making it free, we have to sterilize each and everything. So, on routine purpose, we have to sterilize each and everything to obtain a aseptic area, to obtain a area in which we can work with pure cultures, we can work with that is microbes, but that must not get contaminated from air, that must not get contaminated from the personals, that must not be contaminated from the equipments or glasswares. That is why sterilization is required for each and everything. So, aseptic technique, aseptic area is the area which is free from microorganisms. So, as we need to make our area free from microorganism, we need to do sterilization. Now, what we actually mean sterilization, that the procedures we need to follow to kill the that is microorganisms within the environment or on the surface, even if they are there within the raw materials, if they are there within the glasswares, we have to kill those microorganisms, we have to follow the conditions of sterilization. Now, how we can give those, how we can do the sterilization? There are actually two techniques by which the sterilization takes place in pharmaceutical product. As we are dealing with pharmacy part, so we will talk about only pharmaceutical products. So, pharmaceutical products are being sterilized and being prepared, whatever parenteral sections, mainly we have to see that parenteral products are need to be sterilized, parenteral pharmaceutical products which are being given directly to the human beings through injections. So, they go directly into the systemic circulation. The products which are being given orally like tablets or capsules, that can be prepared within the normal labs. But when we talk about the parenterals, then parenterals have to be that is sterilized, as they are going directly into the systemic circulation. We are injecting those chemicals within the blood. So, we can't take a risk in that case, we have to sterilize those products, we have to use only the sterilized parenteral pharmaceutical products. For that, what we need to do? We have to as we do not want any type of contamination within those products, we have to do the sterilization, and sterilization can be either terminal sterilization or in process sterilization. Now, what is the difference between terminal sterilization and that is in process sterilization? Terminal sterilization is a sterilization in which we do the end product sterilization. Means as you can see even in this, that this is a drug product, this is a container, this is a excipients. Whatever we are going to do in terminal sterilization, we do each and every process in the normal area. Normal area that is that may contains few amount of microorganisms or dust particles, but in this case we always do the end product sterilization. Means once your product has been prepared, whether it is a dosage form, whether it is a liquid dosage form, whether it is semi dosage form, once you have formulated it, once you have that is prepared that formulation, and after preparing that formulation you have packed that formulation within the containers in which you want to dispense. That containers like ampoules, vials, that is single dose containers. We usually use single dose containers in case of terminal sterilization. That mainly terminal sterilization is done on is for end product sterilization, and end product sterilization can only be done if our raw materials which we are being using or which we are going to add within the that is dosage form, that are not affected by the heat, that are not highly thermosensitive, they will not metabolize when that is they will be heated. So in that case only we can go for terminal sterilization, we can go end products. Once the formulation, drug product, excipient has been formulated, has been that is added together, they are added to the container in which they need to be packed, and once the product is whole product has been that is packed within the container in which we need to dispense that product, after packing we go for sterilization product. That means drug product sterilization means once you have prepared, you have after preparing you have added that into the container in which you want to sell that product, and after adding to that container you will keep that product within the autoclave, you will go for autoclaving and you will sterilize at the end. But what we call as in process sterilization, in process sterilization what we see that first if we have drug product, then we will first sterilize the drug product. Maybe it is there that drug product we have to sterilize it in a different way, container have to be sterilized in different process, closure have to be sterilized in different process, excipient need to be sterilized in different process, and after sterilization all the sterile drug products, sterile containers, sterile closures, sterile excipient have to be moved towards the aseptic region, and in aseptic region they will be packed, and after packing they will be dispensed. There will be no end product sterilization in case of in process. If we are going for in process, then there will be no end product, because it may be there that drug get degrade if we are using a autoclaving technique or we are heating the product at the end. So it may happen that the chemicals which we have added to the product, they may get react or they may produce metabolites. So end product sterilization is avoided. The process in which end product sterilization is avoided, that is known as your in process sterilization. After that comes, now why we need that kind of aseptic area and why we need that of kind of a sterilization, because aseptic area we can formulate that is as I told in process sterilization. In process sterilization can be carried out only in the aseptic area. Aseptic area is the area which will be free from microorganisms. So in aseptic area we need to design that area if we want to work in the microbial labs, if we want to work in an industry in which we have to prepare parenteral products. So production of sterile products should be carried out in a clean environment that will be free from microorganisms, that will be free from dust particles. So that is why aseptic area is actually required in the manufacturing industries in which we are going to prepare our parenteral formulation, in which we do not want any type of contamination of microorganisms. So in clean environment we have to prepare those pharmaceutical products, and that clean environment is being divided into 4 grades depending upon the guidelines which government has given. Based on those guidelines, we can classify our clean area depending upon our pharmaceutical industries. We can classify our clean areas into 4 grades. That grades are known as grade A, grade B, grade C and grade D. Now these areas, these clean areas defined. Now out of these grades, grade A is known as your aseptic area. Now that grade A, in which there will be no contamination, there will be not even a single microorganism will be there, that area will be called as aseptic area. There will be no dust particle in that area. Your whole, that is parenteral sections, as well as your if you see your microbiology lab, your microbiology lab is also being divided into these 4 grades, that is grade A, grade B, grade C and grade D. Grade D is the area which is highly contaminated, which will be having lot of microorganisms, which will be having lot of dust particles within it. But as we move from grade D towards grade C, grade C will be having less number of dust particles as well as less number of that is microorganism. Grade C will from grade C will move towards grade B, and grade B will be having very less number of microorganism, or these all limits have been given by various guidelines. We can find them within the pharmacopoeia, that clean area classification, what actually, because all the layouts are being designed based on these classification, these clean areas. So now what are these grades? Now grade A, grade B, grade C and grade D, we can specify these grades. As you can see, pharmacopoeias have mentioned the limits of particles which can be there within these areas. Now we see grade A. In grade A, if we have dust, if we consider a particle which is having a size of 0.5 micrometer, if any dust particle is having a size of 0.5 micrometer, that particle can only be in the range of 3520, that is 3520 particles are only allowed within grade A, whereas as you can see in grade D, there are this area is highly contaminated with the dust particles which are having size 0.5 micrometers. So in this case, as we move from this area towards this area, the number of particles, as you see, are decreasing from 3 lakh or 330 lakh towards the 3000. So in this way, the number of dust particles which are allowed within the grade D area are much more as compared to grade A. Grade A is called as the aseptic area, in which minimum number of particles are allowed. If we see 5 micrometer size dust particles, in that case only 20 particles are allowed, not more than 20 particles. Not more than 20 particles are allowed within the grade A, whereas we can see in grade D there are 29,000 particles may be there of 5 micrometer size. So as in this table, this will be clarified that grade A is the aseptic area which is having less minimum number of dust particles, which will have that is minimum number of even microorganisms. This table is based mainly on the particles, that is dust particles, maximum dust particles which are allowed within that particular area. And next table, if we see, this table is mainly based on the microbial monitoring, that how many microbes are being allowed within the grade A, within the grade B, grade C and grade D. Grade A will be having the minimum number of contamination. As you can see, there will be less than 1 CFU, that is colony forming unit, not more than 1, that means nil. There should be no microorganisms within the area which is known as aseptic, grade A. Whereas if we see for grade D, they can contain even 200 colony forming units within 1 metric cube. That is, in this area 200 particles, 200 microorganisms may be there which can form a colony forming units within a metric cube. So, these are the main techniques how we can collect the samples, like air samples. We can go for settle plate techniques, we can go for contact plate techniques. What we need to do is how we can monitor microbes within the environment, in which we are working, how we can determine that concentration, that what technique we need to follow. These are all techniques how we can collect the air samples. We can collect the air samples to determine that how much microorganisms are there within that air. We can go for settle plate techniques. We can take the nutrient media, we can take the Petri plates, we can, that is, place the Petri plate within at any of the surface, and what will happen when that Petri plate will be open and will be containing any of the nutrient media. If any of the microorganisms will be present within the environment, that will come into the Petri plate, that will generate colonies, that will that when we incubate that settle plate into the incubator, it will generate colonies, and that colonies will be counted. And if the colonies, number of colonies, if we have placed the plate for 4 hours and after that we have incubated, and if we have found any of the microorganism is there within the Petri plate that is less than 1, then that area is classified as grade A, and if the number is more than 100, then that area will be classified as grade D. The area which will be having large number, more number of microorganisms, that will be grade D, and which will be having less than 1 microorganisms, then that area will be graded as grade A, that is aseptic area, that will be free from microorganism, free from dust particles. So, this table is based on microbial contamination, and previous table was based on particle contamination, that is dust particle contamination. On the basis of how we can categorize our clean area, that is grade A, grade B, grade C, depending upon the number of particles present within the environment and number of microorganisms present within the environment. By various techniques we can analyze, like settle plate technique, like contact plate technique, like glove print method. We can note down, we can do the microbial monitoring, and after finding out how many microorganisms, how many dust particles are there, we can classify, we can categorize that particular area and specify within various grades, whether it is a grade A, grade B or grade C. But for aseptic area, if we have to go for terminal sterilization, if we have to that is do the terminal sterilization and product sterilization, then the aseptic area layout will be different, whereas if we have to go for in process sterilization, in that case the layout will be different. If we have to go for terminal sterilization, like in this case, you can see there will be grade A, grade B, grade C and grade D. Now, this is, if we have seen, this is a plus sign, this is grade D, this is C, and this is D, this is C, and this we can see as B, and here we have A. So, as grade D will be having the highest contamination, D, C, B and A. So, grade D will be having the maximum contamination, that is, if we see, we are outside the lab we have maximum contamination, and when we move from one area to the other area, that is material entry. Here we can see the material entry, here we can see the clean changing areas. When we move from normal lab to towards the aseptic area, the whole lab will be categorized into 4 areas. First area will be, we have to move from D to C. The movement must be in a unidirectional way, that means we have to move from D to C, C to B and B to aseptic area. We can't go from A to D directly. Well, what we need to do, we have to first go to the microbial lab. In microbial lab, you have to clean your hands, you have to put on new lab coats. In when you will move from D class, in which there will be that will be a highly contaminated area. When you are there in that area, you have to move to the C grade. In C grade you have to change your lab coats, you have to change your shoes, you have to clean your hands. So you have to take personal hygiene when you are moving from grade D to grade C. And when you move from grade C to grade that is B will be much more less contaminated, that will be less contaminated as compared to D and C, and that is why this plus signs are indicating that is pressure which has been being created within those areas. So this is called aseptic. As you can see, in this case terminal sterilization is occurring, that means we are preparing our solution, that is equipment which are being used for the that is preparation part, that equipments are there within the lab, and once you have entered to the aseptic room, this is the aseptic room, and in aseptic room you are doing the packaging of the products, we pack our products, whatever products, this is a material entry. After having the materials, we go to the compounding areas. In compounding areas, equipments are there for the preparation, and from that preparation will be passed on to the aseptic area. In aseptic area you will pack the products, and after packing you go for the terminal sterilization. There will be an autoclave placed within the aseptic area, means terminal, that is once you have packed your product you have to do the terminal sterilization. So way of moving a person, way of moving a material entry will be same in this layout. If you can see this layout, in this layout we have preparation area, we have that is personal area, we have that is compounding areas are there, equipment areas are there, and also we have the aseptic area, but compounding or preparation is being done at the end in the aseptic room, and after packaging we have to terminally sterilize our product. Whereas the layout of in process, that is totally different as compared to the terminal sterilization. In this area the material entry will be different. Sorry. So this is that is terminal sterilization. And what will happen in case of in process sterilization area? In case of in process, what will happen in that case, that layout, what you need to do is you have to keep two entries, different entries for the materials, you have to keep different entries for the raw materials, you have to keep different entries for the that is personas, means material entry will take place from this area. Here you will enter the material, and here you have to add more area, that is known as, that will be divided into 4 zones. Now 4 zones will be, this is D, this is C, this is B, and this here you will be having the A, but here we will be having the personal entries. Person entry will take place from this side. That is material entry will be from this side, and person's entry from will be from this side. They will not move from one area to the other. The whole area will contain different personas, and personal, this is known as persons will enter from this side and will remain here, and then they will move towards this aseptic area. They can't move to material entry zone, they can only remain within this area. They will exit out, if they want to exit, they will exit from here only. They can't that is come from this area towards this area. So that layout is totally different for that is for in process sterilization, means personas will move from gray area, that is known as your highly that is most contaminated area, that is D what we call as, also we call it as black zone. Black zone means it contains lot of microorganisms, there will be lot of microorganisms within that area. So from black area they will move towards the gray area, from gray area that gray area is known as C, and that gray area will contain less number of microorganisms as compared to the that is black zone, and the from gray area persons can move from gray area towards the white area, and that white area will actually contains no microorganism, will contain no that is dust particles. That area is also actually known as, that A area is also known as aseptic area, means that will be free from microorganisms. Whenever personas are moving from D towards A, they have to take extra precautions that for the personal hygiene. They have to change, there will be a changing room, they have to change their lab coats, they have to put on the, that is hair mask, they have to put down the mask cover, they have to cover, and even their shoes they have to cover with the help of that is covers. Sterilized covers you need to use when you are moving from D towards C, and from C to B. Extra precautions need to be taken so that we can prevent our product from microbial contamination. So, this is actually what we call as that is we have studied about, that is sterilization part, and how we can design the aseptic layout based on terminal sterilized products, as well as we have covered the that is in process sterilization, that how the layout may actually differs from for terminal and in process sterilization case. That movement will be different for materials, and movement will be different from for the personal. In case we want to go for the in process sterilization, if there is no end product sterilization, in that case we have to take extra precautions, we have to take extra surface hygiene measures, we have to take extra personal hygiene measures, so that we can prevent our product from microbial contamination. So, aseptic, this was all about your design of aseptic layout. So, thank you so much.